Good evening, everyone. I hope you can hear me in the back. And welcome to the Arnold and Mabel Beckman Center. I am Frank LaFurla, and I am the Hannah and Francisco J. Ayala Dean of the Ayala School of Biological Sciences. And actually, we are honored to have Hannah and Francisco Ayala here, our namesakes. So if you don't mind saying anything, let's welcome them. I have to say, I am very honored tonight that we are partnering with the Allergan Foundation, and we as a school are exceptionally fortunate to have such a great community partner in Allergan who helps sponsor these outstanding lecture series. I know there are a lot of scientists in the audience here, and I know that many of you work on a variety of disorders, whether it's tuberculosis, and I know Celia's here, or epilepsy like Tally works on, or stroke like Steve Kramer works on, or Alzheimer's disease like many of us work on. The one thing that we all have in common is that whenever we go out into the community, almost everyone asks us, when is the next drug going to be approved for this disease? Everyone is incredibly interested in that. And so before we get to tonight's lecture, I think we should give a round of applause to the amazing staff in the Office of the Dean who helped put on this wonderful event. You know, it's because of their hard work and dedication that we're able to put on these types of lectures that really appeal to the public. And so I don't want to take up a lot of time here, but I have the privilege tonight of introducing Dr. Mitchell Bryn, who is from Allergan, and he has been involved in the therapeutic development of Botox at Allergan for about 16 years. And I think if you came to the last one, maybe 2 times ago, he gave an outstanding presentation about the medical uses of Botox. And I think we'll have to do a rerun of that sometime in the near future because it was so popular. But in addition to his role at Allergan, he also holds an appointment here at UCI in the Department of Neurology as a clinical professor. And I can tell you that I personally am quite honored to have him serve as a member of our Dean's Leadership Council. And it's because of that interaction, and he's been such a great champion of the Ayala School, that we found out about tonight's guest speaker. So with that, please join me in giving a warm welcome to Dr. Mitchell Bryn.
So thank you. It's really a pleasure to be here. I want to just spend a minute about the Allergan Foundation. The Allergan Foundation for probably close to 2 decades has been providing funds for this forum. And the goal is to have a mixed forum and lectures which can address issues which are relevant in a way that both the academic scientists and the community at large can understand. And in that spirit is the reason why we have Rusty here tonight. Now, I've known Rusty for a fairly long time, and I'll tell you a little bit about that in a second. But Rusty did his medical degree at Albert Einstein College of Medicine in the Bronx. He did his internship in general surgery, then at Jacobi, and then went back to Albert Einstein to do a neurosurgery residency, and then a residency in neurology. Rusty then joined the FDA in 1983 in the Division of Neuropharmacological Drug Products. And I'm supposed to ask Rusty to tell us how he got the job. But at this point, I'm going to be a little extemporaneous and tell you a little bit about what I know about Rusty and what I respect so much. I've been down to the FDA multiple times, both in my academic activities before I joined Allergan and as a member of Allergan. And there are a couple of very special things about Rusty. Rusty mentors people, whether they're his staff or whether they're people in the pharmaceutical industry. And he always imparts wisdom. He tries to explain how drugs are approved, why they're approved, and there's this balance between benefit risk and try to bring that to life for us. In addition, he's been very active in the academic community in addition to his activities at the FDA. And another thing that makes Rusty very special is that he will lecture in forums outside of the regulatory environment. And so the American Society of Experimental Neurotherapeutics, he was a regular member and lecturer and participant, and also in the epilepsy community. And so in that capacity, Rusty has moved up in the FDA and ultimately oversaw the entire division of neuropharmacological drugs. And that's a major position in the government. So Rusty would actually have the final responsibility in the neurological environment. So those are drugs that affect the brain or the peripheral nerve. In terms of the decision, do they get approved or do they not? Or what are the questions that one needs to ask a sponsor, whether it's a pharmaceutical company, which it is in most cases, or the academic community? And so in that environment, I've gleaned a lot of respect for Rusty as a communicator and a leader and as a mentor. We're very fortunate that when Rusty retired, he moved to Dana Point. So we've gotten to know each other in a different environment. And Rusty's going to talk with us to give you some perspective on how drugs are approved at the FDA and what that process is. So with that, Rusty, if you would join us.
Thanks, Mitchell, for that very nice introduction. A couple of things. One, the quick story about how I got my job at the FDA. I finished my residency. We moved down to D.C. because my wife Sharon is here. Got into law school, and I was looking for a job. And the only job I could get was doing insurance physicals. And I was traipsing all over the Washington, D.C. area, and I was out at one of the but I had seen an ad in the New England Journal for the job at the FDA, and I was out doing one of the insurance physicals when the director at the time of the neurology group called my house because I wasn't home. So he spoke with Sharon for about an hour, and I think she said, "Look, he really needs a job." So I'm not exactly sure what transpired in the conversation, but I know that I subsequently got an interview, which was a very pro forma thing because I think I had already gotten the job by that point. So, thank you. Second thing, Mitchell said I did a neurosurgery residency. I did 2 years of a neurosurgery residency, and I got out of there as soon as I could. So, that was, we don't have the first slide, I guess, yeah, okay. So, it's a pleasure to be here because I think people know very little about what the FDA does, how the FDA thinks, what the rules are. There are rules that we have to follow. By the way, there's a very good chance I'm going to mostly say we when I talk about the FDA. I don't speak for the FDA anymore, but it's hard to break that old habit. And I may also, if I use the word FDA or agency, I'm really going to be just talking about the neurology group at the FDA that I worked in for 30 years because I don't really know how the other groups regulate drugs and how they think about things. The rules are the same for everybody, but they're very flexible, and how you study a drug and how you think about approving a drug for Alzheimer's disease may be very different from how you think about antibiotics or drugs for cancer. But anyway, I think there's very little known about how the FDA works. It's sort of shrouded in mystery. And even people who work with drugs, even sometimes work in the drug industry, who we deal with all the time, don't really necessarily have a good understanding of how we think about things, which is unfortunate because obviously there are a lot of scientists here who may be working on drugs or drug mechanisms or disease mechanisms. All these things are critical when we think about approving drugs or developing drugs. But those, and of course, many of the rest of us take drugs, prescribe drugs. So I think it's useful for people to know and not for the agency to be shrouded in mystery. But again, it's unfortunate that people don't know. In fact, I have a nice picture of the FDA's new beautiful campus in White Oak, Maryland. So I actually, it looks like this. So what I hope to do here, I've set myself sort of a fairly straightforward task, which is first to talk about what the rules are, what the laws are, the statutes and the regulations that flow from the law, and then mostly to sort of discuss how we think about deciding that a drug is effective. I'm not really going to talk very much at all about safety. That's obviously a critical part of drug approval, but I really want to focus on effectiveness for a couple of reasons. Number one, a drug has to be effective in order for it to be approved. It doesn't matter how safe it is, it has to be effective. So that's critical. The sort of the sine qua non for approval is effectiveness. Also, there are rules that have to be applied when you think about effectiveness. And there are very, very, very few rules, formal rules, for assessing safety. So in some sense, effectiveness, while being critical for approval, is also, it's sort of more straightforward. And there are more rules. The rules are broad. You'll see some of the rules. But nonetheless, there are rules. There are a pathway, and there's sort of bright lines about determining when a drug is effective or not. So I really want to focus on that. What does the FDA do? To my thinking, it does it's engaged in two large areas of effort. One is drug approval, or actually, let's say drug review, because not all drugs are approved. So, this is based on a review of a document called the New Drug Application. I realize there are many people here who may know a fair amount about this, but I gather there are many people here who may not know very much about it. So when a sponsor thinks it has enough information to get a drug approved for marketing, it submits an application called a New Drug Application, or for other types of products, a biologics license application. But basically, I'll be talking about NDAs. They're more or less the same standard for approval. Companies, it's usually companies, will primarily design the studies and conduct them, or usually at academic centers, but not necessarily. And these studies could be performed by many, many centers across the world, multinational studies, multi-center studies. They could be 50, 60 centers. But anyway, they submit it under a New Drug Application. But before that, all the work that's done before in people with investigational drugs is done under something called an IND, an investigational drug application. And every time a sponsor wants to do a study in people with an investigational drug, I'll define an investigational drug as a drug that hasn't been approved in the United States for any use, the companies have to submit a plan and information to support the plan. A plan for a study in people is called a protocol, and there's a fair amount of information that has to be submitted to support the plan that sponsors want to do in people. These include animal studies to look at the toxicity of the drug, to see what organs might be damaged by the drug, so we know how to look for it in people, the chemistry of the drug, a previous human experience if there is any. So that's the IND process. I put it second here. It really comes first. That's where all the data is accrued in humans to support a new drug application. It takes up most of the FDA review time, actually. Everybody focuses on new drug approval, but the investigation of these drugs to accrue the information to support a new drug application takes much more of the agency's review time. So I wanted to show you a little bit how the FDA is structured. I went online and I looked up organizational charts for the FDA. And what I found was basically an organizational chart of the Office of the Commissioner. I don't really know what they do there, but this is all Office of the Commissioner. Drug review is done way down here in something called the Center for Drug Evaluation and Research. And there are other centers. Now, my group is not the only group that looks at drugs to treat neurologic disease. There is the Center for Biologics Evaluation, and they look at vaccines and cell therapies and gene therapies, that sort of thing. And I really don't know. We had no contact with them, and I don't really know how those products are regulated. There's also the Center for Devices and Radiological Health. They have medical devices, and some of them are for neurologic disease. But the vast majority of products that are on the market to treat neurologic disease came through and come through my group, which is located in the Center for Drug Evaluation. I saw this, all of this, and I got this feeling. I thought we were here. But apparently we're over there. But anyway, that's where the drug development is done, in the center. And so here's an organizational chart of the Center for Drug, the Center for Drug Evaluation and Research. And we are, or my division was here in the Office of Drug Evaluation 1, and there are review divisions, as we call them, that cover the entire field of medicine. So all products are somewhere in here. And my group was in here. I just point out there are a lot of other experts who are intimately involved in drug development. There's the biostatisticians. There's the clinical pharmacologists. They look at how the body handles a drug when it's ingested or injected. The folks here analyze the data for the trials. There's a whole group. There are many groups, obviously, that are involved in drug review. The Office of Medication Error Prevention was a favorite group of mine. They ran detailed analyses of whether or not there was a good chance that there were going to be medication errors based on often the brand name that's proposed for a new drug. And does it look like another? Does it sound like another drug's name that's on the market? I once turned a drug down solely because there was very good evidence that there were likely to be medication errors. And that apparently was rare, because the head of the organization, the head of that group, came to me and said, "Do you really want to do that?" Because I don't think people had done that before. But that's at least a preventable error. You can't prevent every bad thing that happens with drugs, but you can try to prevent medication errors. So this is the organizational chart for the structure that I was in, the Center for Drug Evaluation and Research. All right, so let's go over the rules that govern basically the efficacy standard, and how we set out the rules for how we determine efficacy. You know that Congress passes statutes, and the president signs them, they become law, and then the individual agencies write regulations to sort of flesh out, to put some meat on the bones of the law, because the law is very broad. The regulations are broad too, but not as broad. They're a little more specific. So the law is very flexible, can be applied in many different settings, but it's law and then regulation. So the Food, Drug, and Cosmetic Act passed in 1938. There was a tragedy where patients who drank an elixir of an antibiotic, which was dissolved with a substance that was highly toxic. Hundreds of people died as a result of that, so the government decided it was important that the FDA assess whether or not a product was safe before it came on the market. And so this gave authority to the FDA to oversee the safety of food, drug, and cosmetic before they were put on the market. Another tragedy happened in the early 60s when pregnant women took a drug called thalidomide in Europe, not here, which was a sleeping pill or an anti-anxiety pill, or then was touted as a morning sickness pill, and hundreds of babies were born with very, very severe birth defects. So Congress decided maybe it would be a good idea if the FDA had the authority to decide that drugs also worked before they were marketed. So this was these amendments that included the standard for effectiveness, or requirement for effectiveness, are referred to as the Kefauver-Harris amendments in 1962, named after the two Congress people who submitted this amendment. And so now for the first time in 1962, drugs had to be shown to be effective prior to approval, and the standard was what's called substantial evidence of effectiveness. And I'm going to talk about this a fair amount later, but just so you know, prior to 1962, there was no requirement that a drug be effective when it was approved. There are those of you here to whom 1962 may seem like ancient history, but for the first 10 years of my life, when my doctor prescribed a drug for me, I couldn't be sure, and I'm pretty sure he couldn't be sure whether it was actually going to work. So it's not that long ago. So, but there were many drugs approved between 1938 and 1962, and they had no evidence of effectiveness. So the agency decided to go back and look at all those drugs and see whether they really were effective. So they gave the task to the National Academy of Sciences to look at all these drugs and see whether there was evidence to show that they were effective or they weren't effective, or what the story was. So they looked at over 3,000 drugs, give or take, with I think about 16,000 different indications. And they broke down into categories: effective, not effective, and needs more study. And I think about 1,000 drugs were taken off the market that had been approved prior to 1962 because there was no evidence that they were effective. The IND rules were published also in 1962, and this laid out in fair detail about how INDs were supposed to be reviewed, what sort of information was supposed to be submitted to the agency in order for the agency to decide that a study may proceed, and that was a very, very important regulation that was passed. In 1992, there was something called the Prescription Drug User Fee Act, which the agency had always had in the law a requirement to review drugs in a certain period of time. I think it was 6 months, but very rarely did that happen. So in 1992, a law was passed which said that companies had to submit a user fee, at the time $100,000 every time they submitted a new drug application. With that money, more reviewers would be hired at the agency. And that caused a little bit of a stir at the agency at the time. I was there at the time because we're supposed to be regulating the industry, sort of the cops, if you will, and the industry is paying our salaries. So there was some heartburn about that, but nonetheless, it happened, and it's been reviewed every year since then, or periodically since then. I think the current user fee for a new drug that has never been approved in the United States before is somewhere around $2 million now. And I think the agency last year, or some recent year, I think, received over $200 million in user fees. And with that, the agency has been extraordinarily expanded, and now, but deadlines are imposed. And in my experience, 95% of the time, those deadlines are now met. 10 months for most drugs, or 6 months for other drugs. So in 1992, they also changed or added to the law about a different type of outcome measure that could be the basis for a finding of substantial evidence of effectiveness, so-called surrogate markers. I'm going to talk about that in a little while too. But just so you know, that happened in 1992 in the wake of the AIDS disaster. In 1997, another law was passed, or an amendment to the law. This added yet another new definition of substantial evidence of effectiveness, which I will talk about as well. And it also introduced a program for pediatric studies, which I'm not going to really talk about much. And hopefully, whatever I don't talk about here, or if anything needs clarification, I hope there'll be time for questions at the end, because I'm going through this fairly quickly. And then the surrogate marker as a basis for approval was added to the regulations in 1992, which do have the force of law, but they were added to the law, the statute, the FD&C, the Federal Food, Drug, and Cosmetic Act, in 1997. And that just sort of solidified it. I'm not going to talk about these pediatric rules. Suffice it to say that if you're developing a drug in adults, and which is the way most drugs are originally developed, and and that disease that you're developing it for also occurs in children, you have to submit evidence in children for that indication. Anyway, so just very briefly, I don't want to go into this in any kind of detail, but the IND process, it's a years-long process. It's interactive. The agency and the industry meet, and if they don't meet, there's a lot of correspondence that goes back and forth. You may know it proceeds in phases. Phase 1 is usually healthy volunteers, very small doses, just to see if it's tolerated. And then subsequent phases, longer exposure in healthy volunteers, then patients are exposed. Phase 2 is sort of the early clinical studies that look at effectiveness. Phase 3 are sort of the definitive studies, more and more patients exposed as time goes on. And sponsors design the studies. Initially, they do conduct the studies. The FDA doesn't conduct studies. There are often important nodal points where the FDA and the sponsors get together. They talk about the design of the pivotal so-called clinical trials, the clinical trials that are going to serve as substantial evidence of effectiveness. And there's a lot of give and take about the designs of those studies. At any point, the FDA may stop the development of a drug under an IND, mostly because of safety reasons, and there are specific rules about this, but sometimes also because if the studies are so poorly designed that they can't answer the question, the agency can stop a study for that reason too, although that happens very, very rarely. In my division, I think we probably stopped in the early stages of development, about 30% of studies, mostly because there was findings in the animals that the sponsors weren't adequately monitoring people for. In the very early, in my division, we were very conservative early on. Before much or anything was known about a drug other than animal studies, we were very conservative about making sure the studies were done safely, and that's why about 30% of the time we didn't permit them to proceed. Once we got to the end of the NDA, or when sponsors submitted an NDA and we knew the drug was effective, we were very, very liberal, and you'll see some examples about approving drugs that we knew worked, even if there were significant side effects. So you can think I was talking to somebody about this during the presidential primary. So early on, think Ted Cruz. Later on, think Bernie Sanders. And I think that's probably a fair way to do it. So what is a drug? There's a legal definition. It's sort of commonsensical. It's something intended for use in the diagnosis, the cure, mitigation, treatment, or prevention of disease. And also, an article that's intended to affect the structure or function of the body. So the point here is that while most drugs that I saw were developed for diseases, it doesn't have to be for a disease. You can develop a drug to just improve normal memory. That can happen. We rarely saw that, but that's possible. So here's the standard of effectiveness that the Kefauver-Harris amendments added to the law. Substantial evidence the drug will have the effect it purports or is represented to have under the conditions of use prescribed, recommended, or suggested in proposed labeling. So substantial evidence, that's the key phrase, for what's claimed for it in the label. The label I'm talking about is not the label on the bottle. The label is a so-called package insert. If you get a drug from the pharmacy, it's a long, thin piece of paper, and that has all the information that the FDA thinks that the prescriber needs to have about a drug before they can prescribe it. The dose, the pharmacokinetics, in other words, what do we know about the blood levels? Is the drug metabolized in the body? The side effects, if there are bad side effects, there's a section in labeling that highlights those. So all the information the prescriber needs, that's the label we're talking about. So anyway, so it goes on to define substantial evidence. So this is what it means. I put the part of the definition that's important in bold because there's other stuff in there that nobody understands. So, but anyway, the term substantial evidence means evidence consisting of adequate and well-controlled investigations, including clinical investigations. I'll skip the rest, on the basis of which it could be fairly and responsibly be concluded that the drug will have the effect that's in the label, the proposed label. So a couple of things that are important here. Clinical. Clinical means human. So you have to show that your drug works in humans. There's one exception that the agency a little while ago changed the law to add a standard for approval for poisons. And that can be, in particular nerve gas, and that can be studied in animals. It's very difficult to do an ethical study of nerve gas in people. And my division was the first division to use that mechanism to approve a drug for a pre-treatment for nerve gas exposure. So anyway, so clinical means human. Investigations is plural. That means typically you have to have at least two. Congress didn't say you had to have a preponderance of evidence. You just had to have replication, which is sort of a standard scientific thing. It's not good enough if one person does it, somebody else has to do it too. Okay, and that could be concluded the drug will have the effect that the sponsor proposes to be in labeling. So that raises the question, can anything get into labeling? If you do a study of something and you show something in people, will the agency automatically approve it even if we don't know if it means anything clinically? Well, the answer is no, and there was a court case in 1986 which had to do with the agency having removed some drugs that were designed to treat tissue injury. And the sponsor made the case that, well, we did studies and it shows statistical significance. In other words, it met the statistical standard. I want to talk about that too. It met the statistical standard for approving a drug or for showing that it works. So the agency should allow it to be approved. And the court said, the fact that the drug and not chance can be assumed to have contributed to the fact the measure doesn't necessarily establish that the patients will receive a benefit. It'd be anomalous to hold that drug manufacturers may demonstrate effectiveness merely by showing statistical significance. So this is a court saying it's really up to the agency to decide whether or not the drug should be approved. And statistical significance by itself isn't adequate. Now, the agency was doing this anyway, quite frankly, but this was an important case. Although very few people, I think, at the agency know about this case. But this is standard. It's up to the agency to decide if a drug has a clinical benefit. It's not enough just to have statistical significance. So then in 1997, they added an additional benefit, a definition of substantial evidence. So if the secretary determines, based on relevant science, that data from one adequate and well-controlled study and something called confirmatory evidence, that could be sufficient to establish substantial evidence of effectiveness. So I said investigations was plural, and for many years, that was the only requirement, the only standard. Now it says, so sometimes one study is good enough. But the vast majority of drugs are approved, at least in my experience, on the basis of at least 2 studies. This one study standard is applied, but not usually. It's atypical. And we can talk about what the standards are for one study if we have time later. Anyway, so the regulations, so this is adequate, the law says adequate and well-controlled investigations. The regulations flesh out a little bit the kinds of studies that would qualify as adequate and well-controlled. Placebo, concurrent control. The bottom line about the trial is that there has to be a control group. In other words, you have to compare the patients who get your drug with some other group of patients. And there are many different types of controls that you can employ. But you got to compare it to something, because if you just give your drug to your patients and they get better, maybe they would have gotten better on their own. So we don't know. You have to compare it to something. So I'm not going to go into great detail this for time reasons, but the placebo control, concurrent control means you take patients, you randomize them to either your drug or placebo in inactive treatment. You compare how they do. Sometimes you'll have dose comparison, which is patients are randomized not just to one dose of your drug, but let's say to multiple doses. Dose X, dose 2X, dose 4X maybe, and you can also have placebo there as well. A no treatment concurrent control, sometimes you really don't have to give them something else, but you do have to follow some other group that isn't treated. Active treatment concurrent control, I'm going to talk about that a little bit later, where you compare your drug to another drug. There's a lot of interest in showing that how a particular drug compares to another drug that's already approved for that same indication. So that's an active control trial, and then historical control, where you compare the patients who get your drug to some other group of patients that aren't even in your study, some other group outside that you have data on who are like your patients, and you compare them. These are the 5 types of studies that the agency defines in the regulations as potentially adequate. They're not all adequate for any given situation, but they can, under certain circumstances, be adequate and well-controlled. The vast, vast majority, if not all the studies that I dealt with over my tenure at the FDA were placebo concurrent control. There were often dose comparisons, so you could have your drug, 1 dose, 2 dose, and placebo, but almost all the studies we looked at were placebo-controlled studies. They're the most efficient and the easiest to show an effect if your drug actually works. The basic elements of a typical study. Clinical trials can be very complicated. The way you analyze them can be very complicated, but the reality is fundamentally, they're fairly simple. You take a group of people who have the disease you want to study, you randomize them, you put some get your drug, some get the control, usually placebo. You have certain inclusion criteria. They can't be too sick. They can't be too well. You want to be able to show that the drug has an effect, and at the ends of disease severity, it may be difficult to do that. They're double-blind. In other words, the patients don't know what drug they're getting. The doctors who are giving it or who are evaluating them don't know what drug they're getting. And then, very critically, you have to have outcome measures and a way to analyze it. And you have to say this up front. Sponsors measure lots of different outcomes, but you have to pick 1 or 2 at the front and say, this is what I'm going to rely on. I'm going to test this, and if this is positive, and I'm going to talk about what positive means, but if this is positive, it's a positive study, and if it's not positive, it's not a positive study, it's a negative study. And we often get into discussions with sponsors where they're the outcome that they picked as primary failed, didn't meet the statistical test, but some other outcome that they did, which they now like better, was positive. And there can be arguments about it, but the rules are very simple. You say what you're going to do up front, and you say how you're going to analyze it. There are many different ways to analyze data. You have to pick a particular statistical method. You analyze it with that method, the outcome you said you were going to do, and that's how you decide whether a study is positive. And there are often arguments about, well, we picked the wrong statistical method, so we really should have analyzed it this way. And when, of course, you do that, it turns out to be positive. But that's really not allowed, except under rare circumstances. So outcome measures, this is a critical part of the clinical trial world. You have to measure how you decide whether or not the drug is having an effect. So the important considerations, at least the way we thought about it, was that it had to matter clinically. In other words, you had to, apropos of that law case, you had to be measuring something that mattered, that actually helped the patient. And it had to help the patient. We had sponsors come in and have outcome measures that were directed at the caregiver of the patient. And we hadn't allowed that. It's an interesting thought, but we said that it had to matter to the patient clinically. And very often, and I'll show you some examples of types of outcome measures, but very often we didn't really know what was an important treatment effect. When I first got there, the standard for approving a drug for epilepsy was a 50% reduction in seizures. So if you had half as many seizures on the drug as you did before the drug, that was considered a win. Well, 50% decrease in seizures is very nice, but what about 40% decrease in seizures? That's good too. So we don't really know in the typical case what an important effect is. So we like to have the outcome measure itself embody something that is clinically meaningful, measure something that's clinically meaningful, so that if you win, if you have any change that is statistically significantly different from placebo, that's good enough. So that was very important for us in terms of what we allowed as or relied on as a primary outcome measure. We didn't really care too much about effect size. So there are many different kinds of outcome measures that we relied on. So for example, you could count events. In epilepsy, we just count seizures. How many seizures do you have in a month? That was the outcome measure. Migraine, the proportion of patients who didn't have a migraine or whose headache went from very severe to gone. And multiple sclerosis, this is a disease characterized by neurologic events. It could be anywhere in the nervous system. We just counted how many events you had over the course of the study. So we just count things. Those are on their surface clinically meaningful. Any decrement in a bad event, we thought was good. So there are many types of scales that are multi-item scales where you rate patients. They can rate themselves sometimes, or the caregiver rates them, or a physician rates them. But either way, there are many scales, and many drugs are approved on the basis of effect on scales. So Parkinson's disease, a movement disorder, there are standard scales that you measure. In ALS or Lou Gehrig's disease, there are standard scales, and these are multi-items, and you just look at the average score on the scale on drug versus placebo, and that's how you win. Restless leg syndrome, patients feel odd sensations in their legs. They feel the need to move. There are scales that you can assess the severity, the frequency, that kind of thing, and you just look at the difference in scales between drug and placebo. Sometimes if scales can be very, very sensitive, they can pick up very, very small things, and we don't know, if it's so small, we don't really know what that means clinically. So we say, in that case, you have to win on the scale, but you also have to win on some sort of global measure of functioning to ensure that that change that you saw on the scale actually meant something to the patient. So there you have two primary outcomes, and you have to win on both of them. There are time-related outcomes. In Parkinson's disease, there are episodes called off episodes where patients are very stiff and they can't move. You say the time you spend in that period. Insomnia, how long it took to fall asleep. How long did you stay asleep? In multiple sclerosis, we can sometimes look at the time till they got worse in their disability. So there are many time-related outcomes. They're all acceptable. So there's a new type of outcome measure they talked about in the law, which is a surrogate marker. Surrogate marker is not a clinical. I've been saying something has to be clinically meaningful. Here's a case where it doesn't have to be clinically meaningful. This is a lab test or an imaging, an X-ray or an MRI that really the patient can't feel, but that it's believed that an effect on that is reasonably likely to, and here's the law, it's reasonably likely to predict the clinical benefit. And we can talk later about what situations you would do this in. The neurology division has never relied on this. And there was some heartburn in the agency when this was proposed, because it says it's reasonably likely. Every other outcome we've been talking about is a manifest effect on something that's clinically meaningful. This says it can only be reasonably likely to predict the clinical benefit. And this is intended only for very severe, serious treatments where there isn't much available. But these surrogate markers are becoming very, very popular, and there are problems with them. This, for example, was a study done in 1991, which looked at 2 drugs which treated arrhythmias, abnormal heart rates, which patients had and which patients died from. Well, so this was a study which looked at drugs, these 2 drugs, which had an effect on these arrhythmias, and looked at these drugs on people who had recently, more or less recently, had a heart attack. And it was being used off-label for that. And it turns out that these drugs actually did decrease these abnormal heart rhythms. It decreased the arrhythmias, but it also increased mortality. Patients died more frequently. So this was proposed as a surrogate marker. Let's just measure these arrhythmias, these abnormal heart beats. But in fact, even though these drugs benefited those, they hurt the patients. So there can be a dissociation between the effect on the surrogate and on the clinical outcome. There's another example I'm going to skip. So clinical trials, are they a perfect reflection of the real world? No, they're not. The patients who are enrolled, we talk about inclusion-exclusion criteria, are a very highly select subset of patients who actually have the disease. They typically have fewer other illnesses. They typically take fewer medications than everybody with a disease. As I said before, they're not too sick, they're not too well. So they're a very highly select group. And the clinical trials that the FDA relies on to establish substantial evidence of effect on this really can be thought of as a proof of principle. In other words, the drug works in some group of people who have the disease, but we don't know if it works in everybody necessarily. Because the samples in studies are highly select, they're not particularly representative entirely of the people who have the disease. So all drug development is compromise. And I don't mean just with the drug company. There certainly is compromise with the drug company. Drug company wants to study a drug for this long, and we say, no, it has to be this long. Or they want to use these doses, and we say, no, you have to use these doses. And you come to a compromise. But that's not what I'm talking about. It's all compromise because you can't do the ideal study. Ideally, the drugs I dealt with were generally given for a patient's lifetime, or at least for many, many, many years. But you can't do a study that lasts 10, 15 years. So if you think there's a safety issue, let's say you think from animals the drug might injure the liver. Well, ideally, you'd like to monitor the function of the liver every day. You'd be able to pick something up early, but you can't do that. So we never have really definitive information about how a drug works. It would be nice to have that, but it may be 15 years after the drug is approved before we really find out exactly how the drug is working. So it's all compromise, and if it wasn't, there would be no drug approval. So I think that's very important. So why can studies be positive? They can be positive because of fraud. People could make up the data. It's very rare, but the agency, if a company submits a drug for a new drug application, the FDA goes out and inspects some of the sites that perform those studies, and they look at the data, at the original data, and they look to see how it compares with the data they actually submitted in the new drug application. Fraud, in my experience, is extremely rare. Bias. One group, the group that gets a drug, might be somehow different from the group that gets a placebo, and that could be affecting the outcome as opposed to just the drug. You want the groups to be as perfectly the same as possible, and you want the only difference to be the treatment and the placebo. So how do we deal with bias? Well, typically we review the studies to make sure that the studies are adequately designed. Very often the randomization takes care of a lot of this. It ensures, to the extent possible, that the 2 groups that you're dealing with are exactly the same. And blinding, so that the physicians don't know which patients are on drug because they're going to be rated differently if they know they're on drug or placebo. So bias is another way to deal with it. Chance. It could just be chance. And the way we handle chance is statistics. I'm going to talk about that in a little bit. And then it could be drug effect. If you rule out everything else, then the drug did it. That's the whole effort here, is to figure out, did the drug cause the difference, or did something else cause the difference? All right, so I'm a little hesitant to do this because this is statistics, and nobody really understands that. So, but it's a lot of different ways to analyze trials. But in the neurology division, there was one type of method that was always used. And the output of this method, after the statisticians do what they do, is something called a p-value. Now, I know a lot of you talk about p-values. It's in every scientific paper, p-value less than 0.05. Many of those p-values are inappropriate, and they don't really mean what people think they mean. But nonetheless, p-value is the output. So you get all this data, thousands of data points in a clinical study. You give it to the statistician. They go in a room, and eventually they come out. And they come out with a p. We don't know what happens in that room, but they come out with a p-value. And I'm going to try to explain what a p-value is. So the p-value is the probability that the difference that you saw between the drug and the control group, usually placebo, was due to chance if the true state of nature is that the drug and the placebo are identical. So you assume going in that the drug and the placebo are the same. In other words, the drug doesn't work. You do the study, you get the p-value. And if the p-value is less than 5%, let's say, and that's the typical standard, we say that the study is positive. In other words, so we say 5% is a very small chance. It's very unlikely that it was due to chance if the p-value is 0.5, if the reality is that the drug and the placebo are the same. So because it's a very small chance, we say the drug and the placebo are not the same. Our assumption that the drug and the placebo were the same is wrong, because this was highly unlikely due to chance if that was true. So that's what a p-value is, and you have to do it twice typically. So that just means that the odds that the result you saw was due to chance if the drug and the placebo are the same are extremely small. So you say, well, the drug, therefore, our assumption about the drug and the placebo being the same is wrong. We reject that assumption, and this is the standard. Now, I'll have to say, you will not find an official agency document that says the p-value of 0.05 is how it's referred to, is still 5%. A p-value of 0.05 is the standard for drug approval. It's not in the law, it's not in the regulations, it's not in any formal official FDA document that I know of, but it's tradition, and this is the standard. And if your p-value is 0.1, you lose, generally speaking. So this is a bright line standard. It's in all the medical literature. This is what a p-value is. Has to be 0.05 or less, and that's the rule, and everybody understands that. But again, there's nothing official about it. It's tradition. So what are some characteristics of studies that support effectiveness? Well, they're short. As I said, most of the drugs that we dealt with are given for a lifetime or many, many, many years. But typically, studies are 3 months or 6 months. For epilepsy, 3 months. For Alzheimer's disease, 3 months or 6 months. So it's very, very short compared to how long the disease is going to be treated for, and that's a matter of practicality. We don't really know whether these drugs work out in time beyond what we study. Studies only demonstrate an effect on symptoms as far as we know. We know very few drugs, if any, that actually affect the progression of the disease. And certainly, unfortunately, we don't have any cures yet. There might be some of the multiple sclerosis drugs might affect the underlying disease. The agency has never said that officially, but that might be true. But as far as we know, the drugs we approve really only have an effect on symptoms and not the progression of the disease. And people are trying to figure out how to study progression of the disease. There's no requirement to show that the drug is as good as or better than other drugs. We talked about active control, you compare it to another drug. There's no requirement for that. And in the neurology group, rarely, if ever, was there a showing or an attempt to show that a particular drug was better than another drug. How a drug works isn't particularly important. This may sort of seem counterintuitive, but we don't even understand how the drugs work. If we waited to understand how a drug works, as I said before, it would take years and years to approve a drug. What we care about is, does it work, and is it sufficiently safe? That's what counts, not what the proposed mechanism is. There was once a case, before I got there, called the navel drops case. A company came in and wanted to study a drug for hemorrhoids, and they applied the drug through a dropper in the patient's navel. And so it's ridiculous. So, the division who was dealing with that said, no, you can't do this study. That's ridiculous. And the company appealed that to the head of the Center for Drugs, and that person said, of course they can do it, if it can be done safely. We're not smart enough to know that this isn't going to work. It didn't work. But the point is, you don't know in advance. We're not smart enough to know in advance whether or not a drug works, and so it's not really an important part of deciding whether or not a drug is effective. As they say, every drug has three activities: the ones you want, the ones you don't want, and the ones you don't know about. So today's ridiculous idea is tomorrow's Nobel Prize, and there are examples of that. So we don't really care. Not all patients need to be helped. Very often in a study that we consider positive, lots and lots of patients aren't helped. Only a small fraction are helped. We don't know, we're not smart enough yet to know who, in most cases in advance, is going to respond to the drug. But people are very excited about personalized medicine, but it hasn't really come to neurology yet. Not all symptoms need to be treated. Diseases have many, many symptoms, but not all of them have to be treated in order for a drug to be considered effective. The effect of the drug can be small. And on average, unfortunately, the effects for neurological drugs in general are relatively small. But that's good enough. And patients don't have to get better at all. Every patient in a study of an effective drug may get worse. But they get less worse than patients on the placebo, and that's good enough. So, for example, we can see here an effect on symptoms. These are all, I'm going to show you some examples from approved labeling. It's all public. So here, patients get better. This is a drug for Alzheimer's disease. So they get better on a scale, but then you see they start to come down. This is the placebo here, and this is the drug at 2 different doses. So they get better. That's an effect on symptoms, but ultimately the disease progresses. So that's fine with us. So active control studies, we talked about comparing 2 drugs. People are very interested in how my drug compares to some other drug. Most of those studies are done to show that the new drug is as good as the old drug. It would be great if it was better than, but that's a hard thing to show. So here's an article from the Annals of Internal Medicine a few years ago by my then boss. Now these compared 2 antidepressants. This imipramine, well studied, it's been shown to be effective in many, many studies comparing it to placebo. And it was compared, these are, I guess, 6 studies which all compared this other drug, which is a more toxic antidepressant, nomefensine, to imipramine. So they also started out here on this scale of depression, and after the study, these drugs were all the same. Or these 2 drugs were the same. So that's good. The problem is these studies also had placebo in it. And as you can see, in 5 out of the 6 studies, the placebo did exactly the same. So that's the problem when you do studies of an active control study that doesn't show a difference. We can talk later on about why this is an issue. But if you didn't have placebo, you'd say that nomefensine was as effective as imipramine. So that's the problem. The mechanism of action, as I say, is unknown. This is reflected in product labeling. Every label that comes out of the Division of Neurology, at least when I was there has to be a section that's called mechanism of action, but everyone starts out the same, which is the precise mechanisms by which this is a seizure drug, produces its analgesic and anabolic actions unknown. And then you sort of put in what you think you know about the drug. Maybe it's relevant, maybe somebody's interested. And so this is all, it says what we think it's doing, and then at the end it says, however, the relationship of this binding to the therapeutic effects is unknown. So you want to tell people we don't know. We don't know. And this is a drug that was just approved for ALS. I wasn't involved in this because it just happened. And here they didn't even bother to give any theory. It just says the mechanism is unknown, which is, an honest statement. Not all patients are even enrolled in studies. Here's a Botox. The indication for Botox is the prophylaxis of headaches in adult patients with chronic migraine greater than 15 days per month with headache lasting 4 hours a day. Very specific. This isn't just a treatment for migraine. This is for people who've had at least 15 headaches, 15 days per month with a headache and lasting 4 hours. So you see, it's a very restricted group of patients with migraine. Alzheimer's disease is standard. Alzheimer's claim mild to moderate dementia. Typically, we don't know if it works in severe dementia, at least when these drugs are initially approved. Restless leg syndrome, indicated for treatment of moderate to severe primary restless leg. So these are these are subsets. Not every patient is evaluated. Not every patient is helped. This is another outcome measure in an Alzheimer's study. And this is a sort of a global measure, and you can see these are the ratings. And let's look at the people who were improved. So the black line is the drug. That's the high dose. Let's look at the black line. So it's about 20% of patients were minimally improved, moderately improved, and markedly improved, maybe another 5%. So maybe 25% of patients in this study were improved. That's fairly common in an Alzheimer's study. And everybody else either didn't change, about 40% didn't change, everybody else got worse on the drug. Now, they got less worse than the placebo patients. That's good. But many people in the study weren't helped, and that's sort of standard. Small effects, we look at small effects. This is that drug that was recently approved for Lou Gehrig's disease. The difference on this scale, I forget how many points in this scale, but these patients got worse. They got less worse than the placebo, but it's a very small difference, a difference of 2 on this scale. So it's a very small difference, but that's good enough. Here's another study of an epilepsy drug. If we look, let's just look at the black. These represent different doses in one study. So about the biggest difference between drug and placebo at the high dose was about 17%. That's one seizure that you didn't have that you were going to have on drug. That's good enough for us, but 17% is a pretty small difference. I'm going to skip some of these just for time purposes. I want to point this out. This is a drug for epilepsy. One study, the patients who got the drug had 1,600 seizures per month, and on placebo they had 700 seizures per month. And at the end, there was a 26% reduction in the patients who got the drug. So 26%, and this is a 5% increase, so it's about a 30% difference, about 26% reduction in seizure. That's pretty good, but that still means they had 1,200 seizures per month. But anytime you can decrease the number of seizures, that's a good thing. This is, I know we have some Allergan people here, but this is a change from baseline in the frequency of headache days. It's not a big difference between drug and placebo, but it reached the statistical mark, and it was plenty good to get approved. Here, I just want to make a point here, and by the median change, that means half the patients got more of this change, and half the patients had less of this change on thumb flexor muscle tone. That's a small movement, but it was important, and it also worked on finger flexion. It also works on many other muscles. But the point here is that this is a very small movement, but it was very important for patient care, for patient hygiene, for the patient's hand to be kept in a good shape. So they didn't have to be able to pick anything up or type or anything. It just they had it. That was important enough to approve the drug. There's a box warning. I don't want to go into this. Here is a drug for epilepsy, and these patients had 8 to 9 seizures per month before drug, and at the end of the study, they still were having 5.5, and there's a difference of 2 between drug and placebo. That's pretty small, but it's good enough when we're talking about epilepsy. These are bad diseases. And this drug, so that's a pretty small effect. This drug has a box warning, which is the most significant warning you can give. This is a drug that causes, in some people, the permanent visual loss. Not complete loss, but it starts as constricting the peripheral vision. That's very bad, and in most people it's irreversible if it happens. But that's the treatment effect, at least in one study, that we approved, even in the face of that significant toxicity, because these are bad diseases. These are bad diseases, and this is that box warning. So these small effects on average, but for any, we used to put these sorts of graphs in all the labels, then we figured out nobody understood them, so we took them out. But here, just to point out, so this represents the the drug group, and this is the placebo group. So even though these drugs have small effects, what this shows is that for any given change, and this is change in this direction is good, but so this means that this number of patients on drug had a 3-point or better change. So that's like 60%, whereas on placebo, it's very small. So what this shows is that even though the average effects can be small, for any given degree of change, a large change or a smaller change, there's always more of those people on drug got that change than the people on placebo. So, and this is just another example of that in an Alzheimer's drug. Patients don't have to improve. This is an outcome measure that looks at time to disability, and this is and this shows the difference between drug and placebo. It took longer for patients on drug. There were fewer patients who reached disability in the course of the study than on placebo. That's not a huge difference, but disability is an outcome. As I said before, patients can get worse. And this is another example. I'm going to skip this because of time. And finally, this was the drug that was just approved for ALS. And not only do patients, not all patients have to improve, but sometimes no patients improve. And that had that as you saw from the average scores earlier, that they were they progressed slower than the patients on placebo. But if we look here, this is the number of patients who had any particular degree of worsening. 4 to 5 points, 6 to 7 on this scale. It doesn't matter what the scale is. So everything to the left of this arrow means patients got worse. So in this study, everybody got worse, with rare exception. These are the people who got better. That's a couple of percent, 2 or 3%. So not everybody has to get better, and sometimes in some studies, nobody gets better. So just in summary, and I think I've gone over, but so there are laws and regulations that govern drug development, and particularly with regard to effectiveness, as we've been talking about, but they're very flexible. And so the agency can approve a drug even with small treatment effects if we are convinced that those effects are real, the drug did it, and it wasn't a chance finding, or it wasn't related to bias. And many of the drugs that we approved in neurology had pretty small treatment effects. We waited in vain for a company to come out with advertising for the new drug to say, drug X, we're better than nothing. But it didn't happen. But the point is, on average, many of these drugs are not that much better than nothing, meaning in comparison to placebo. But the diseases we dealt with were bad. Even migraine, which doesn't kill anybody, can be excruciating. It can be life-altering. So we accepted a lot of toxicity, as long as we understood what the toxicities were and could put it in labeling and warn people about it so they could decide whether to do it, even in the face of relatively small treatment effects on average, because we dealt with diseases that were bad, and anything that helped patients even a little bit was good enough for us. So I think I'll stop there.
So we have time for a few questions from the audience.
Yeah. Well, I don't know the 21st Century Cures Act in excruciating detail. I do know that there is something in there about a patient experience. It has to be taken into, or real-world experience has to be taken into consideration. I find that to some extent troubling. This is my personal opinion. I don't know how the agency is going to think about this, because personal experience or real-world experience, whatever that is, generally speaking, at least with the diseases we deal with, it can be highly misleading. I'm not even sure what they mean by that. You know, if a particular physician has treated 30 patients and they seem to have gotten better, it's not data. You know, it's anecdote, and as a general matter. So I worry about that. I actually, again, this is a personal view. I have faith that the agency will look at that data because I think they're probably going to have to, but I think they will give it its just due. So that's what I'm mostly concerned about, and I think that's the way they'll think about it. As far as personalized medicine, you still can do adequate studies with regard to, genetic subsets and that sort of thing. So I think good science can be applied and will be. Well, no, you can take people with a certain genotype and you can randomize them, and you see whether the drug works in them or doesn't. Oh.
Hello, my name is James Pham from Westminster High School, and at first I'd like to thank you for your presentation today. And a question I'd like to ask with a bit of background before I ask it. As a student athlete and aspiring bodybuilder, the FDA is kind of a gateway for our supplements and stuff. For example, sometimes stuff slips through your safety net, like Jacked or Jack 3D as some of us know it, which has a banned mental stimulant. But some drugs, such as HGH and decadurbalin, have a lot of benefits for the human body and drawbacks too, but let's not worry about those. But they're tightly locked away because of those drawbacks, and as a result, some's not even manufactured in the United States anymore. Decadurbalin, you have to go out to other countries to find it. So, as an officer of the FDA, how many times?
Ex-officer.
Yeah. Okay, close enough. But so, as an officer, how many times have you seen or halted, like actually came in and halted, like usually a drug? How many times have you usually halted one or seen one halted? Like
Well, when you say halted, do you mean a drug that someone is trying to sort of bring in from another country for their own personal use?
Or like developing currently? Like how many times have you had to stop one?
Well, as I said, I think people keep statistics on this kind of thing. So when I was talking about the IND process, my division stopped development of an investigational drug, I think somewhere around 30% of the time. Mostly because there was some, as I said before, some signal from animals' work that there was a real problem and there was no good way to sort of figure out if humans going to get it. And the vast majority of those come off, this is called a clinical hold. The vast majority of those drugs come off hold because the sponsor goes back and gets the information they need, or they change the clinical study to monitor patients better. I think that's what we did. We weren't afraid to stop it, but it's always a failure of something if you have to stop a study. You mentioned supplements. There's a whole other law that regulates how supplements get on the market, and there isn't much to it. I mean, there's a law called the Drug Supplement, I think, a Health Education Act or something, but it basically, Congress said for supplements, whatever those are, like those are the things you get at the GNC or vitamins or, coenzyme Q or other stuff, they can come on the market. They don't have to have any information about whether it works. They cannot make disease claims, but they can make what are called structure function claims. As I said before, they could say, improve your memory or improve your athletic performance. They can say that. They can't say it treats Alzheimer's disease, although some do, but that's illegal, and the agency doesn't have that much capacity to go after every drug or supplement that makes an illegal claim. Very often on TV, you'll see some ad for a supplement, and the bottom in letters that you cannot read, it says, these statements have not been approved by the FDA. So, a lot of us in the drug development world weren't too happy when that happened, but it's the law of the land. I'm not sure I answered your question.
Yeah, you did.
Okay.
Looks like HGH is not coming to the mainstream market anytime soon. So,
No, well,
thank you.
again, there's all sorts of illegal drug use, as you know, but no, they would have to study they'd have to pick an indication. And by the way, I didn't say this, but you know, drugs are approved. They're not just approved. They're approved with this very specific indication, and they have to have evidence to support that indication. So if somebody wants to develop it for a particular indication, they could do it. They could do it, but they have to go through the process.
All right, let's take a couple more questions. And down back there.
Doctors that the NSAIDs have been revealed to be much more harmful longitudinally. Yeah, I'm repeating it, that NSAIDs like ibuprofen and naproxen, et cetera, are more harmful than over the long term than was thought by some. Why aren't longitudinal studies, ongoing longitudinal studies, part of the approval process, where approval is given conditionally as long as the continued research is done?
Yeah, no, that's a good question. Sometimes it is. Sometimes there are things called post-marketing requirements that the agency can impose when they approve a drug. And so we say, you're approved, you can be on the market, but you have to do the following studies. And if there is a reason to believe that there is a problem or will be a problem, you get sort of hints of it. You know, we didn't talk about safety, but the safety data that you have at the time of approval in the typical case is relatively meager. And in terms of duration of exposure, the basic rules, the minimum rules are 300 to 600 people for 6 months and 100 people for a year. Now you can amend that, you can change that if there's a reason to change it, but that's sort of that's the minimum. It's not that much. So, but if there's a hint that there's an issue, you can require sponsors to do anything post-marketing, have a 5-year study, or a 10-year study, or a pregnancy registry, or anything. But it isn't. I mean, in my experience, that wasn't a routine requirement. And you could argue, why not follow every drug that is approved for as long as you can, right? 10 years. It's a good question, and I don't know what the agency's current thinking is. I know they have thought more about chronic safety since I left, but I don't think that's a requirement yet. And you know, we have some folks from industry here; they may be interested to weigh in on that as well. But it would be an interesting idea. I don't know if it's a cost issue but it's a good idea.
So let's take one more question. I think you had a question. Yeah, want to wait for the microphone?
I'm wondering how much of a bias is introduced into the drugs that you review just by the fact that it costs $2 million for the application. For example, we have a big opioid drug crisis right now, and there are people with legitimate pain issues, and yet others, you know, there's a hesitancy to treat them on long-term on opioid drugs. Some people have suggested that marijuana might help alleviate pain with some people, and yet that's not a federally allowed drug to be used. And I can't imagine someone paying $2 million to put in an application and whatever costs for a big clinical trial to be approved by the FDA for that kind of use.
Well, look, that may be. I mean, it may be that if something is freely available, it's very difficult to get people into trials of it if they can just get it, particularly if they can get it legally. And you know, that's an issue. You can't. The FDA doesn't really have the authority to force somebody to submit a new drug application. We don't have that authority, so it's really up to the sponsor to decide.
Can you waive the fees?
You know, that's an interesting question. There are some products for which the fee is automatically waived, like orphan diseases, a disease that has a prevalence of 200,000 or less. There are fewer than 200,000 people in the United States who have that disease. That's an orphan disease, and you don't have to pay the fee at all if you're an orphan disease. But for a regular product that isn't an orphan, I'm not aware that the fee can be waived. Again, there are folks in the industry, maybe they have some experience with this, but I'm not aware of a case in my area anyway where the fee was waived. That fee of $2 million is much less for other types of applications, but for a new drug, a drug that hasn't been approved before yet as a drug in the United States, I think it's around $2 million now. I don't know if you know if Mitchell, a fee can be waived.
let's do one last question. Amy, you'll get the last question here.
Okay, I get to have the last word. So given that some of these drugs can have a very small effect and yet be very expensive, I imagine you've thought about how if the FDA is not going to decide this sort of cost-benefit analysis thing, how do we as a society figure out how to pay for this, right? So, my husband's a Kaiser physician, and there was a drug that was approved by the FDA that doesn't really do much, but now Kaiser is being pressured to provide it. So how do we deal with that?
Yeah, no, that's above my pay grade. No, I don't know. I mean, it's not an FDA question. You're right, the FDA doesn't have the system that they have in Europe where they approve a drug and they also, I gather, approve the price. I don't know. Well, certainly, and again, Yeah, no, right. I mean, I have no expertise in this area, but there are, the paying, the insurance folks and the formularies and this sort of thing, they'll decide whether to pay for it or not. But the FDA, I can't really answer the question. The FDA has at least traditionally hasn't had a role in that. And it's obviously a problem, but the FDA has no authority to decide how much a sponsor is going to charge, and that's either good or bad, but somebody out there has to do it, but it's just not part of the FDA's authority.
Okay, with that, why don't we thank Dr. Katz again for this insider's view. We're gonna give the mic to you.